Okay, so the fat mass and obesity or FTO gene is located on human chromosome 16 and has been positively linked with an increased risk of obesity among other cardiovascular diseases, including diabetes. The gene contains the code for the FTO protein and is also part of the LP family, but is structurally different from other members. The gene is also known to have the highest influence on the body mass index compared to the other known genes, and this can be harmful since it has been linked with an increased risk of cardiovascular diseases. The FTO gene is also one of the biggest indicators of polygenic obesity. Higher levels of RNA demethylation is found in obese people, and the FTO protein is the first known RNA demethylase, which can also correlate it to obesity. It can demethylate 3-methyluracil or 3-MeU in single-stranded RNA and 3-methylthymidine or 3-MET in single-stranded DNA. The FTO protein is made up of multiple binding domains and through which compounds can inhibit the FTO protein through. The inhibition of RNA demethylation caused by FTO is proven to be very vital in order to try to combat obesity and other types of cardiovascular diseases. Compounds can participate in interactions such as the van der Waals forces, hydrogen bonds, or pi-pi interactions, which would allow them to inhibit the protein through. These types of interactions were analyzed along with the binding affinity to find the best compounds that could potentially aid to combat obesity.
So for methodology, we docked a lot of molecules with online tools to further examine, and if they passed our things, we further examined them. We used a guess and check method to start. When a compound with a high binding affinity was found, we examined the properties of the compound, and we tried to derive similar molecules to that. The molecules from existing compounds were made through the online tools such as Avogadro, and they were also docked with the FTO protein and autodock. This helped us find structures that would end in the highest binding affinities. Many molecules shared similar characteristics, which meant they binded well together. Over the course of the semester, many of the properties that made the scaffold more compatible with the FTO protein were found.
Okay, so looking at our results, so first, after going through the process of docking with various compounds with different structures, it was found that compounds that were derived from fission and imodin produced better results. The structures of these compounds allowed them to interact better with the FTO protein, resulting in higher binding affinities. Another pattern was that the compounds with more oxygen atoms also produced higher binding affinities, and the oxygen atoms typically participated in more interactions, allowing them to interact with and inhibit the FTO protein through. The structures of imodin and fission were similar, and they resulted in higher binding affinities than other compounds. Almost all of the derivatives of imodin and fission that were docked and tested using autodock were involved in more hydrogen or van der Waals interactions in the binding domains of the FTO protein. And based on these results, it was hypothesized that compounds that have more oxygen atoms and similar structures to imodin and fission would be more likely to produce better results.
The fat mass and obesity or FTO gene that is located on human chromosome 16 has been positively linked with an increased risk of obesity among other cardiovascular diseases. The FTO protein is known as an RNA demethylase, which can also correlate it to obesity. The FTO protein is made up of multiple binding domains through which compounds can inhibit the protein through, which has been proven vital to combat obesity and other types of cardiovascular diseases. The compounds participating in the interactions were analyzed with the binding affinity to find the best compounds that could aid to combat obesity through inhibition. In the methodology, the properties of a compound with high affinity were examined from existing compounds and the FTO protein using online tools such as Avogadro and autodock to derive molecules with similar characteristics. Many of the properties that made the scaffold more compatible with the FTO protein were found, such as fission and imodin, which resulted in higher binding affinities. An additional pattern showed that the compounds with more oxygen atoms produced higher binding affinities and allowed for more interactions with an inhibition of the FTO protein through.
We would like to thank ASDRP and their sponsors, as well as our advisor, Rhea, for the opportunity to work on this project and perform this research. And then below is the QR code with our references. Thank you.
Very nice job. Kirk. Any questions? We gotta have some questions. How exactly did you design your inhibitors? That's the question. It's in the chat.
So basically, we designed a ton of molecules on the different online tools, and we docked it with the FTO protein to find which one had higher binding affinities, and then we further examined them and try to make molecules with those similar properties to get higher binding affinities.